In behavioral neuroscience, the two alternative force choice task, or 2AFC, is the foundational tool for measuring decision-making. When a subject performs this task, researchers can record exactly two observable metrics: which option they chose, and the reaction time it took to reach that choice. This is a split histogram plotting those reaction times. The frequencies for choice A point upward along the y-axis, while the frequencies for choice B point downward, mapping the speed of correct versus incorrect decisions. The problem is that these static distributions are ambiguous. A subject might produce a fast reaction time because they processed sensory information efficiently, or simply because they acted with less caution. To differentiate those hidden variables, we have to look past the static outputs and mathematically model the decision as a continuous physical process unfolding over time. The drift diffusion model treats decision-making as the accumulation of noisy evidence toward a defined threshold. This equation, dx equals adt plus cdw, formalizes that accumulation. At every time step, evidence is gathered for one of the two choices, with random noise injected into the input. Solving this equation using traditional statistical models presents a severe practical bottleneck. To extract reliable parameters, these models require an exhaustive number of trials from every single subject. Researchers can bypass this limitation by applying hierarchical Bayesian modeling, specifically through the open-source Python toolbox known as HDWM. By combining the drift diffusion model with Bayesian inference, researchers can extract latent cognitive variables from sparse datasets and map them directly to physiological brain states. Understanding how HDWM accomplishes this requires deconstructing the physical architecture of the drift diffusion model. This diagram illustrates multiple drift processes accumulating evidence over time toward an upper or lower boundary. The average speed and trajectory of these jagged lines is the drift rate, denoted as parameter v. The required amount of evidence needed to commit to a choice is defined as the boundary separation, or parameter a. This distance represents the subject's level of caution. Before accumulation begins, there is a fixed delay required for initial sensory perception and motor execution. This is the non-decision time, marked as parameter t. If a subject has a prepotent inclination toward one choice over the other, the starting point of the accumulation shifts. This starting point preference is defined as bias, or parameter z. The exact termination times of these random walks map onto the reaction time histograms generated by human subjects in a 2AFC task. Parsing these four parameters provides a quantifiable fingerprint of the underlying cognitive decision process. In clinical neuroscience and fMRI studies, constraints often limit the time a patient spends in a scanner, restricting subjects to low trial counts. Standard frequentist parameter estimation techniques, like maximum likelihood, treat every subject in the dataset as a completely independent entity. This scatterplot shows the result of feeding maximum likelihood a sparse dataset of just 20 trials. The line representing the point estimate jumps wildly in an attempt to fit the noisy, insufficient data. Mathematically, the model either overfits to the noise or loses the true cognitive signal entirely. Independent parameter estimation is incompatible with the data limitations common in modern cognitive neuroscience. Hierarchical Bayesian modeling provides the structural solution to this sparse data problem. Instead of treating individuals in isolation, the model assumes that individual subject parameters are drawn from an overarching shared group distribution. This graphical node network maps that hierarchical structure. The inner plate containing trial data nests inside an outer plate for individual subject parameters, which then connect up to the overarching group distribution nodes. This architecture introduces a statistical phenomenon known as hierarchical shrinkage. Here, the group distribution acts as an informative prior. It pools statistical strength across the entire population, pulling wide, scattered individual estimates inward toward a single, smooth bell curve. This pooled variance anchors the noisy estimates, which prevents overfitting and maintains accuracy even when a subject completes only a fraction of the standard trial count. To perform this complex estimation, HDDM utilizes Markov chain Monte Carlo, or MCMC, sampling as its computational engine. Rather than producing a brittle point estimate, MCMC generates full joint posterior distributions, providing a rigorous quantification of uncertainty for every parameter in the model. Hierarchical Bayesian modeling rescues sparse datasets by leveraging the shared statistical strength of the population to validate the inferences made about the individual. Simulations using synthetic data quantify the statistical power hierarchical Bayesian modeling retains over maximum likelihood. This chart plots mean absolute error against the number of trials. The upper line tracks maximum likelihood's high error rate, while the lower tracks hierarchical Bayes. A critical divergence occurs between 20 and 50 trials, where HDDM maintains a low mean absolute error. The hierarchical model reliably recovers true cognitive parameters from datasets that frequentist methods would render unusable. This incredible statistical efficiency enables the immediate application of DDM analysis to highly constrained environments, such as live intraoperative recordings. The utility of this model extends beyond static group estimations to dynamic, trial-by-trial physiological data mapping. The HDDM regressor functionality allows researchers to feed external measurements into the model as covariates. One study demonstrated that frontal theta power, measured via EEG, correlates with increases in decision thresholds during high-conflict choices. This animation illustrates how that EEG theta power data feeds into the model. As the theta wave peaks, the decision boundary, parameter A, dynamically expands in response. This regression architecture provides a method to quantify how a specific neural signal regulates the decision-making process in real time. We can test what happens to these parameters when the brain's neural network is physically disrupted. Researchers also administered this experiment to Parkinson's patients undergoing deep brain stimulation of the subthalamic nucleus. This graph plots posterior density distributions of theta regression coefficients on the decision threshold. The green curve, patients with stimulation off, shows a positive correlation, meaning the neural brake functions normally. The blue curve, stimulation on, shows a complete reversal, as stimulation disrupted caution adjustment. Because MCMC sampling generates full posterior curves, researchers can confidently quantify this separation probabilistically, bypassing the need for frequentist p-values entirely. Hierarchical Bayesian estimation of the drift diffusion model provides a rigorous framework for mapping and quantifying the biological mechanisms that drive human choice.